Hello everyone. We'll just wait for another 2 to 3 minutes so that everyone else can join, and then we'll start off with the webinar. Till then, you guys can just tell me whether you are able to see the screen and whether you are able to hear me properly or not.
Yes, we are able to see the screen.
Yes.
Okay, thank you. Tane, can you please confirm whether everyone else has joined in or not so that we can start off with the lecture? It's 3 already. Tane, can you hear me?
Yes, yes.
Yeah, so should we start or should we just wait for another 2 minutes?
We can wait till 3 to 4 minutes, then we can start at exactly 3:05. Will
Okay,
it be
okay,
okay?
sure. Okay, no issues.
Yeah.
Hello.
Hello.
Yes, hi.
Yeah, we'll just start off in a minute. So we're just waiting for everyone
Oh,
else to
okay,
join.
okay, sir.
Yeah.
Okay, sir. Okay.
Yeah, I think Tanya, I think we should start now.
Yeah, sure. We can start.
Okay.
Okay.
So hello everyone. Good afternoon, and I hope you all are doing well in these times. My name is Satyam Sangeet, and today we are going to look at the webinar for next generation sequencing. So first of all, I would really like to thank all of you who are present here and giving your time to this specific webinar. So with the advancement of various kind of methodologies which are out there in terms of DNA sequencing or RNA sequencing, we know that with the involvement with the advancement of technologies, people have also moved on with different kind of technologies to sequence the DNA. Now, the very first thing that comes in our mind is why there is a need to sequence DNA, or what exactly is meant by DNA sequencing, right? And slowly we will progress into seeing how this DNA sequencing is being converted or being upgraded, so to say, in the sense of next generation sequencing, right? So the very first thing that we are going to see is what exactly is DNA sequencing. Because before going in with the concept of sequencing, one has to understand the basic ideology behind how the sequencing happens and what is the need for those things, right? So very first thing is what exactly is DNA sequencing. Now, in a layman terms, if we have to define what DNA sequencing is, just the process whereby we can get to know what specific nucleotides are present in a specific pattern. Let's say ATGC or GATC, right? Different kind of patterns which are out there in terms of nucleotides in a DNA sequence, and to understand how those sequencing is happening and after which specific nucleotide a new nucleotide might enter in the DNA sequence. That is process which is known as your DNA sequencing in a basic layman overview, right? But if we have to understand it from a complex point of view, or let's say from a technological point of view, what we can say is that a DNA sequencing is basically a process whereby we take a specific DNA sequence in order to know what are the different nucleotides that are going to be added when you start the process of DNA sequencing. So as to create a DNA sequence of our own interest or a specific DNA sequence that we are going to target, right? Now, DNA sequence is basically a specific order of nucleotides, right? Whether it is ATGC, GATC, CTGC, whatever, right? So this is very basic, small 4 or 5 nucleotides, but your DNA sequence can be 1,000 or 5,000 nucleotides long as well, right? Now, there are different methods which are out there that basically target the question of DNA sequencing and how one should proceed in this specific manner of DNA sequencing, right? And over the years, this process has grown a lot with the advancement of technologies. So initially we had with us the maximum Gilbert method of DNA sequencing. Then people find out that there are specific drawbacks with that method, so we upgraded the method, and then came the Sanger's method of DNA sequencing, which was more reliable, so to say. And then also there are, we know that not every process is 100% accurate, or not every process can give you what is the desired result. So you have to keep upgrading the process with the advancement in the technology. So you have to keep in check the process as well as the technological advances. Then after that, people figured out that the major drawback with all the classical method of DNA sequencing was that not large amount of data could be sequenced all at once, And that was a real deal for people to understand, because what happens is going with a shorter form of DNA sequences and then sequencing those DNA for a large amount of time is a very tedious job. It's a very labor intensive job. And considering the current situation, people are always in a hurry, right? We need the output as fast as possible. So then came the new advancement, or so as to say, the new generation, which is termed as the next generation sequencing, whereby you can sequence a large amount of data, a huge set of data in a very short amount of time, right? So that is how the advancement in the DNA sequencing, or so to say, the evolution of DNA sequencing started out, right? So, just a second. So next thing is, before proceeding on with the specific, the terminologies involved and the specific processes which are involved in DNA sequencing, the very first thing we have to keep in mind is what are the applications of DNA sequencing? Like, where are the major forces or major areas whereby we can actually imply or actually, use these applications for our benefit, right? So there are various areas where you can actually apply DNA sequencing methods. For example, you have evolutionary biology. So as you know, evolutionary biology is a field whereby we basically look at how a particular species has evolved with the changes that are occurring in the evolution, changes occurring in the environment. And based on those environmental aspects, how the sequencing has been changed in terms of, let's say, mutations, or let's say in terms of the compatibility of the organism to adapt itself with the fluctuating environment, whether be it a fluctuation in thermal conditions or nutritions, or increasing global warming, which increases CO2 productions and all those stuffs, right? So to understand how those things are happening, what people do is that we take the organism, we take the DNA out of those organisms, and then we do the sequencing so as to analyze that over this period of time, let's say 30 years or 40 years, how that DNA sequencing has changed, or the sequences has been changed in terms of mutations. Let's say in a particular place, A changes to G or C changes to T or something like that, which finally allows the organism to have a better adaptation with the changing environment. Or sometimes it is not always a positive effect. Sometimes it might cause a derogatory effect on the organism itself, right? So thereby you have this ability of using the DNA sequencing technology in order to understand the evolutionary background of a microorganism with the environmental conditions, right? Then you come to molecular biology, so which is a very vast field. And people, when we talk about DNA sequencing or RNA sequencing, people think that these are the process that are mainly involved with the molecular biology field only, but that is not the case, right? So molecular biology definitely is a field which would like implement a large amount of DNA sequencing data, or we can say majorly DNA sequencing is involved with molecular biology. But we have different aspects as well, whereby you can actually try and apply these methodologies as well. So in molecular biology basically consists of understanding every process from molecular terms. So let's say if you are looking at an organism's phenotypic change or a genotypic change, and looking at it from a molecular point of view, like the very basic point of view, like the minute things that you want to look at, then that comes under your molecular biology field. Next thing is your virology. And now virology has taken up a large interest among all the academics as well as scientific field, because with the current situation that we know we are all going through. So you have to have certain technologies that allow you to sequence the data so fastly and so accurately that it saves you enough time, as compared to all the wet lab experiments that you would do. Let's say initially, if people would go for a normal DNA sequencing method, like just strictly using the classical methods, then it would somewhere take around them, let's say for example, it took them around a month to sequence something, and that too, you have to understand whether the sequencing that has been done, whether that is accurate or not, or whether the results are really beneficial for our use or not, right? And then let's say if those data are not well or not suited according to your need, then all that 1 month of endeavor would go down the flush, right? So nowadays we have to have this technological advancement in order to accurately as well as fastly develop a method or to sequence something. And very main thing is the virological sense, because considering the current situation and how the health sector is getting deteriorated with the advancement of viruses, we have to have a basic understanding of how those processes take place in virology as well. Another aspect is medicine, whereby medicine is also evolving in such a sense that nowadays, for example, let's say if you are having a fever, or let's say if you are having a cough, you could just take a paracetamol or combiflam, and that is a very broad spectrum of antibiotics that you take out to make yourself good or feel good. But what people started thinking was that why not we create medicines for a specific person's use? Because what happens with a broad spectrum of antibiotics is that for, let's say, person A, it shows a good effect, but sometimes it's not always necessary that a medicine will always have the same effect on every person, right? So everyone has a different sort of reaction to that antibiotic. So people started figuring out what should be the next breakthrough that we can take place in medicine. And that's when the terminology came as the personalized medicine. That is, you're creating a medicine for a specific use for a specific person. Although the process is really expensive, not suited for a general population because people can't afford that much amount of money. But that is one breakthrough that we can think about, because let's say if you have this technology which will allow you to sequence the data of a person and then develop specific medicines for that specific person, then obviously we can target n number of diseases according to that, and people would really benefit with that, right? And that's where your sequencing methods also find their application. Another method is your okay. So another method is your forensics. So as you all know, forensics deal with a lot of criminology and DNA sequencing, molecular biological sequencing. So to understand or to have an advancement in technology in terms of forensics, that might provide us a help, because so as to catch a specific criminal in a short amount of time, because we know in today's scenario how long it can take for us finding a culprit based on the classical technologies that we have right now. So an advancement in that forensics field might also give us a boost or give us an edge over taking out the details of the criminals and finding out the criminals way faster as compared to the classical methods that we are sticking to right now. So DNA sequencing and all those next generation sequencing can find an application in forensics as well. And last but not the least is metagenomics. Metagenomics is a very new terms, which is not really that much old. But understanding what metagenomics is basically is that understanding metagenomics is in the sense that you are going to basically sequence the data of an organism based on the environmental cues that you have. And very best example of these things could be found out, because I can definitely say from a research background, there's a research currently going on at MIT whereby people are basically looking at how your prokaryotic and your eukaryotic systems are basically adapting with the change in conditions. Let's say if you have a pond whereby eutrophication takes place, and biomagnification takes place. So these prokaryotic and eukaryotic organisms have a huge impact on our environmental conditions, because they are a major contributor towards the global climatic increase. And to understand how these organisms, these microscopic organisms, are adapting with this change of environmental conditions that is taking place. Let's say if you have a community of organisms that resides in a pond or that resides in a thermal lake, and to understand how these organisms are able to adapt with these sort of things can provide us a very in-depth analysis of the molecular mechanisms which are involved. And then they might provide us an edge over to understand how, as we humans, could adapt with the changing temperature, and what we can do in order to protect our environment from the rising global temperature and all those various stuffs, right? So these are the basic broad domains where you can actually find how sequencing basically helps out. And if you go in much in-depth analysis of these individual fields, you have a whole lot of ocean of knowledge that you can gain in these specific fields. But this is a broad spectrum in which we can categorize the application of a DNA sequencing methods. Right now, the next thing is there are various kind of sequencing methods which are available, but we will start off with the classical methods that were out there before the next generation sequencing came into picture. So DNA sequencing were initially categorized in two senses. One was which was called as the Maxim Gilbert sequencing, and another one was the chain termination method or the Sanger's method sequencing. So we will be looking at each one of them individually, And then we will see like how over the years these methods had optimized themselves in order for us to be presented with NGS right now in our hand, right? So we'll first look at the maximum Gilbert sequencing. So maximum Gilbert sequencing, as the name suggests, came from two eminent scientists. One was Alan Maxam and Walter Gilbert, and they gave the sequencing methods in order to understand how the patterning in DNA sequences happen, right? Now, patterning was what happened with them was they were really interested with looking at the patterns which are out there in the environment, and we know from a mathematical point of view that there are specific formulas that can actually give you a specific pattern, right? Let's say a logarithmic pattern or a Fibonacci sequence. So as to say, there are n number of mathematical derivations out there which give you the patterns of environment. But what they were interested in was to find whether those kind of patterns or similar kind of pattern or different kind of patterns actually exist inside us as well. And that's how they came with the idea of why not sequence a DNA to see how those sequencing actually changes out and how those sequences help us to understand a person much more better from a molecular point of view, from a genetic points of view. So what happens in a maximum Gilbert sequencing process is that basically it is also known as a chemical termination process, because the basic idea behind this is that you use a chemical to terminate the evolution of your DNA, or to terminate the process of your DNA or elongation of your DNA. So you start with a sample DNA. So let's say you have isolated your sample DNA from an organism, which is basically a double-stranded DNA, right? But we know that a double-stranded DNA is way more stable, and you can't, do any kind of changes with that. So in order to start your process, the very first step is that you have to process down your double-stranded DNA into a single-stranded DNA. So the very first step involves converting your dsDNA or double-stranded DNA into a single-stranded or ssDNA, right? So basically, you're preparing a homogeneous ssDNA. Now, once your single-stranded DNA is prepared, now you are basically ready for any kind of your DNA sequencing process. Now, before proceeding on with the DNA sequencing process, the very first thing that we have to understand is the basic idea behind how DNA sequencing happens and what is the basic methodology. So keep in mind that no matter which DNA sequencing process you use, the broad idea or the broad methodology that is involved behind the sequencing process is really the same, right? That is, first you have to create a DNA library, and then you go for the sequencing, and then you go for your data analysis. That is the broad spectrum in which a sequencing happens. And then you can twitch your procedure accordingly to your own need. Let's say you want to twitch your model at the time of sequencing. So people can try different kind of processes for DNA sequencing, right? You can go for Sanger's method for sequencing. You can go for your next generation sequencing. You can go for RNA sequencing. You can go for your chip sequencing. So the broad spectrum or the umbrella under which DNA sequencing happens, you have a broad spectrum in which you can work for. But the individual thing, when you go into an individual steps, that changes according to the need of the researcher or the need of the person that you want the process to go for. So the very first step in this maximum Gilbert process was to isolate your double-stranded DNA and then convert that double-stranded into a single-stranded DNA. Once your single-stranded DNA is converted, or you get the single-stranded DNA, the next step is that we label it. We label it with a radioactive phosphorus molecule, a 5' phosphate group, and we label it at the 5' end. Now, the reason we label it is that because to analyze the data, we go for what we call as the autoradiography, and there we would get a different amount of luminescence or radioluminescence, which will allow us to understand whether the process happened actually occurred or not. So the very first step was we isolated our double-stranded DNA, we converted it to a single-stranded DNA. Then the third step was we added our radioactive phosphorus molecule to the 5' end. Now let's say this is our template sequence that we have. Let's just take this as an example here. Now what happens is you have a cleavage at specific nucleotides. So what we do is that we apply certain chemicals in order to cleave our nucleotide at a specific positions. So what we do is that once you get your single-stranded DNA molecule, you have 4 test tubes with you, right? And in all those 4 test tubes, you put the sample. And then in all the 4 test tubes, you put different chemicals in order to understand whether which kind of the nucleotide base you have there. So let's say you want to go for just guanine. So the type of chemical that you would apply in this particular test tube is dimethyl sulfate, also called as DMS, which is followed by a piperidine. So these two combination of chemicals will only give you the G nucleotide, not the rest of the nucleotides. But you can't apply the same chemical formulation to these all the other three test tubes, because what happens is you want different nucleotides in different test tubes, right? So for every test tube, you apply different kind of chemicals, which will give you a different kind of combination of nucleotides that you want. So let's say if you apply your DMS and piperidine, it will give you a G, and similarly, it goes for different kind of other chemicals as well. Now, sometimes what happens is, let's say we have these 4 specific cleavage sites that you can get for. Now, what this A plus G basically stands for is that, let's say in a test tube, you apply DMS and piperidine, you just got the G nucleotide. But sometimes what happens is, if you change the combination of chemicals, those chemicals might cleave your DNA sequence at G, but sometimes it also cleaves the DNA sequence at A. That is, it is not a specific sequence. It might cleave at A, it might cleave at G as well. And that's why we term that test tube as A plus G, or the cleavage at A plus G. And the same way, you can go for another test tubes whereby you apply a different set of chemicals, which will only give you the C nucleotide. And then you apply a different chemical, which will give you C plus T. Basically, that chemical can give you a C cleavage as well as a T cleavage as well. So now how this cleavage basically happens is that, let's say you applied your A plus G, and then you applied the chemical. So the cleavage might happen at a specific positions. So let's say, just a sec. Yeah. So what happens here is, once you apply this specific chemical, so you can see this is my DNA sequence. Now here is the A plus G cleavage that is basically happening. So how do you interpret this result? Is that, let's say you applied this chemical, then it might cleave your channel. So you can see the sequences are here. So it can first cleave at A here. So you will only get GCT, that is 32 phosphorus, then GCT. If it cleaves here. Also what happens is it can cleave at G as well. So if it cleaves here, then you get what is known as GCT and C. And let's say if you cleave your channel, if it cleaves your nucleotide here, then you get this sequence GCTA CGT. Now what happens is it won't cleave your channel, it won't cleave your DNA sequence from here, because then there's no point, because if it cleaves it from here, then there's no point in obtaining a DNA sequence, right? So it will always leave the terminal nucleotide as it is, and then it will start cleaving out from a specific nucleotide. This is a combination that you might obtain if the cleavage happens at A plus G. Let's say cleavage happens at G, then it can cleave your DNA sequence here, then you can just get a G. Otherwise, it can cleave your DNA sequence at here as well, then you can get a G, C, T, and C. So this is another combination that you might get. Let's say if it cleaves at C, then what happens is, if it cleaves at here, then you get a G sequence. Let's say if it cleaves here, then you get G, C, T, and A sequence, right? And similarly, it is for C plus T as well. If it cleaves at C, then you get a G. If it cleaves at T, then you get GC. If it cleaves at C again, then you get GCTA. And if it cleaves at T here, then you get GCTA, C, and G. So basically what happens is, once this cleavage is done, the reason that we are adding different chemicals to different test tube is that you get sequences of different length over here, right? So this is a 3 sequence length, this is a 5, this is a 4, 7 times. So for in a specific test tube, you would get a different amount of sequence length. And once you